To introduce everyone to the NCBI virus and to create a sort of query that's workable and a useful tutorial query, I'm going to focus on the 2014 and 2015 outbreak of H5N8. To make this query simple, I'm just going to look at the H5N8 sequences. But because of reassortment, this probably, in reality, is not a good idea, and I should actually think more about all of the H5s that occur within those dates, even though they might be from other subtypes, because the H5N8 tends to reassort with N1, with neuraminidase 1 or neuraminidase 2. But to make this query as simple as possible, I'm going to focus on the H5N8 sequences. Now, this was important because H5, the highly pathogenic form, which was all descended from a goose from Guangdong from 1997, had been geographically contained to Eurasia. It started in China and it has slowly, from 1997, moved across into India and Pakistan, and there are some cases in Egypt and North Africa, and also cases in the Arabian Peninsula. But it hadn't really gone over very long distances to either the main parts of Europe or across the Pacific to go into the United States. An interesting feature of many influenza viruses is that they have a different evolutionary history in the United States, in the whole of the Americas, compared to Eurasia. So these provide two separate populations that are, their barriers between them of the Atlantic and the Pacific Ocean prevent them from mixing. If you have a human case, this is not true because people fly in planes and therefore take the diseases across these natural barriers. But for animal diseases, such as avian influenza, this is quite a strong barrier. In 2014, there was a large outbreak centered on lakes in Korea of the avian influenza containing the H5 highly pathogenic form. This combined with neuraminidase 8, which was an unusual subtype combination. It didn't occur very often before then. It was usually with N1 or N2, which with N1 and N2 it's a much more fatal disease, and the disease didn't spread because it prevented the birds from migrating. When it was combined with H5 and N8, they were able to migrate even while ill, and they were able to migrate over very long distances. So they were able to migrate over the Bering Straits and into North America, and they were also able to migrate all the way to Europe. The bird migration, they spend the summer months in the Siberia and Altai and the Mongolian highlands, and then during the winter, when those areas become frosted and permafrosted, so there's no food available, they go to European or to, the flyways connect all the way into the Pacific Northwest of the US, but mostly actually they go down to Australia. It's not actually that common that there's a flight from the Altai to the Pacific Northwest, but some of the birds that are on the Pacific flyways are going to come in contact with some of the ones that are going down the flyways which take them through Japan and down towards Australia. So that's the migratory background. So what we want to do is look at the sequences that occurred in that outbreak so that we can trace their evolution and their geographical location. So this is called phylogeography. Following an outbreak of a disease by looking at where is the original sequence and then watching as it evolves and as it moves geographically from one location to another. So we're tracing the movement of a virus, and we're able to trace it because we can see the evolutionary changes from its ancestral sequence and as it's evolving as it moves. So let's set up the database. Now if you go to NCBI Virus and you go to NCBI Virus Influenza, you have 13,890,698. Now, first thing we want to do is focus on the genotype. So we only want H5N8. So when I submit that, I reduce it down to 8,724. Now the specific sequence that has the high pathogenicity in the hemagglutinin, which evolved in the goose in Guangdong, so I want to concentrate on the segment which is hemagglutinin. So if I go to the segments, I wish to switch on segment 4. So once I've switched on segment 4, which is the hemagglutinin, I'm now down to 1,355 sequences. Next thing I want to do is change the collection dates. Now I'm going to, because I can't remember the exact dates that it started in 2014 or the exact time that it's going to finish in 2015, I'm going to take a window around it. So I'm going to take from 01/01/2014. So I'm going to take from the 1st of January in 2014. And I'm going to take it till the 31st, this month. The 31st of December 2015, the next year. So I'm taking a 2-year sample. So when I combine those, I get down to 196 sequences. Now another thing that I want to make sure that I don't get is partial sequences. Sometimes because of fast sequencing, they don't collect all of the sequence data, they only collect a part of it, the part that makes a unique diagnosis. So I want to look at nucleotide completeness, but I'm actually going to do that by specifying the sequence length. So I want all the sequences that are longer than 1700 nucleotides. The hemagglutinin should be a bit longer than that. I think it might be sometimes 1704 and sometimes 1750, depending on what variability there is in the untranslated regions at either end. So if I put a broad spectrum limit of 1700, that will cover everything. So once I've done that, I'm down to 185 sequences. Now, there's a very nice feature in the NCBI Virus database that I hadn't seen in other databases before in terms of filtering, and this is this ambiguous characters feature, which I like. So this allows you to remove any sequences which contain a large number of ends within the sequence. If the ends are in front of the start codon or after the stop codon, so they're untranslated regions, then probably it doesn't seriously matter. It is a big problem to have ends in the middle of sequences, and sometimes because of this partial sequencing problem, that people did not spend the money to do a detailed sequencing to do it thoroughly and get a complete sequence. They just deposit something which has a lot of ends saying there are nucleotides there, but we don't know what they are. This causes a problem for subsequent analysis in sequence alignment and in phylogenetic tree analysis, where you depend on distances and substitution and scoring matrices, because these things pollute your mathematical calculation. So I'm going to get rid of every sequence that has more than 3 ends in a row. So when I've done that, I actually just reduced it by 1. So now I've got 184 sequences. So now I can download these sequences. I want to download the nucleotides because that's what I've got. I want to download all 184. I want to build a custom accession line. So I've got the GenBank title. There are other things that I'm going to need later so that I can look at the phylogenetic trees and the locations and trace the outbreak. So I need to know, as a separate part of the header, where they are, what species they're in, because this helps me to think about migration and where they might have gone, and the date that the sample was collected. So if I go down at this, I want to look at country. In the US, they also have the US states. That will be blank if it's a country which is not the US. Otherwise, that will be filled in. I want to know the host organism, and I want to know the collection date. So when I've got all of that information there, I can download the data, which downloads to a file called sequences.faster, which I'll go to that folder. I need to now rename that particular file to something useful. I double clicked it and opened it in View, which is a little inconvenient. Don't look at the View one because it's not properly aligned at the minute. Right, to rename this, the date today happens to be the 30th of the 1st 2025. So the way to write it is the year, the month, the day, what the sequences are. These are HA sequences from H5N8 in the Korean outbreak, and the data comes from NCBI Virus. And I've used underscores to make sure there's no spaces, because when I come to run some of the programs, like the alignment programs, if I run them at the command line, if I have spaces, it makes it more complicated to type the commands for actually running the alignment programs. So it's always a good idea to put underscores in the name.